Okay, welcome everybody. Last week of lecture. Man, all right. So, urogenital tract infections, my hopes were to have covered this last Friday, but got a little bit behind. That's okay. We're going to kick up the pace a little bit and try to get through this and into 24. It's a little more detailed with the GI tract stuff. I want to spend a little more time in there. So, with urogenital tract infections, there are numerous players. Sometimes we think about normal flora setting the stage for like urinary tract infections, E. coli, and some others can be big players. And then we will think about some other opportunities with sexually transmitted infections, stuff that shouldn't be normal flora. So there are lots of opportunities when we think about a urinary tract infection. In the lab, you have worked with inoculating loops to spread and transfer the bacteria. And my gut says every time you roll in there and grab an inoculating loop, they probably all look a little bit different and bent and all sorts of weird, right? Sometimes they work pretty good, and sometimes they maybe dig into the agar plate. That's not what we really want. When we think about urinary tract infections, we want to try to plate them out and have the ability to look for individual colonies and get some details. So these loops are different. Typically disposable loops, plastic loops, and the loops, oftentimes they're double-sided. Both ends of it are loop, one bigger and one smaller. And the smaller loop looks like it will hardly hold anything. These are calibrated to deposit a single microliter of sample. And you know the volume you have plated. And as you spread that out onto the plate, you can start counting up colonies. A handful of colonies might be expected, and it truly depends on the type of urine sample that you are presenting with. But sometimes we would expect to see a little. When we start approaching, handfuls, you get 50 and above, then we start wondering what's going on, potential culprits. All right. So this media is selective for specific organisms. I'm seeing all sorts of things on here, and I searched through. I'm like, what is this plate? It is a selective plate, but the actual name of it, I haven't found it yet. I did see a lab manual that was talking some of this stuff with urinary tract infections. And right now, off the top of my head, I can't remember that plate. When I look at this, I feel like I'm seeing multiple things on here, like the potential maybe for some lactose fermentation. That's what I feel like with these pink guys. And then the others where we see these darker pigments, and I feel like we've got multiple things streaked out on here. I don't know for sure, but I feel like this reminds me of like when you guys did the Sims Deeps and you potentially saw the tubes turning black as a result of hydrogen sulfide production. That's what I think about with this. All right. Whether or not that is accurate. I'm not sure. All right. E. coli and potentially Proteus mirabilis and vulgaris are both possibilities for some of these UTIs. So I feel like maybe that's who we're looking at. Doesn't matter. So a quick anatomy with this, with the urinary tract, we think about truly more of a sterile environment from the bladder moving up into the kidneys, but the potential for organisms to colonize and maybe take hold and cause issues and moving up the ureters into the kidneys, resulting in greater concerns are there. There are definitely differences in the potentials for this dependent upon male or female. And that sets the stage for greater likelihood, some of a proximity lends to greater opportunities for females to get some urinary tract infections from the result of normal flora like E. coli and others. So cross section of the kidney and moving into it as we look, we're taking a blow up of one of these lobes. And as we blow this up, you can see the glomerulus. And we talked before with those immune complexes like Streptococcus pyogenes causing glomerular nephritis. This is right where we're impacting, right within the glomeruli, and it gets massive inflammation as a result of those complexes linking up to this, to the surface, and potentially causing destruction. We're looking at this like it's one little bit, but this lobe is full of glomeruli. It's full of all of these capillaries moving through here to filter the blood. All right. So it looks like it's just one thing in this, but it's chock full. All right. So as we're circulating through, filtering, and the potential for organisms to make their way up and cause issues are there. So both male and female reproductive system, and you can see some potentials lending to maybe greater likelihood with females to ultimately contract some UTIs as a result of that. We'll go through some details as we move through here for some potentials for STIs shortly. All right. So these urine dipsticks, it's not the same as a pH strip, but it's pretty similar when I think about this stuff. And you can get pH on there. Our bottom bit right on here is pH. But we're looking for other things, looking for proteins, cells, any sort of abnormality. And somewhere on here, maybe right here, is our control. And we look for deviations with that to clue us into some aspects of that. Before we've ever thought about plating it out, we can test with the dipstick and get some details respectively. So all of these deviations potentially lending to the hints of infection, and then plating it out and truly trying to identify it, and hopefully finding some sort of narrow spectrum antimicrobial to deal with that, to not set the stage for some secondary issues. This next organism's spirochete right here, Leptospira interrogans. This is a bacterium that we put under the umbrella of a zoonotic infection. It is pretty common to see in the urine of different animals, and coming in contact sets the stage for us to potentially get infected. So I recall years back a commercial talking about a little kid that contracted Leptospira from playing on a playground, and dogs doing their business, or maybe even cats in the sand, and the urine ultimately harboring these organisms and setting the stage for issues like that. This one, I'm not sure if our urea plasma was the organism on that plate that we started off with or not. Maybe, but I also feel like that was a little bit different and it looked more like it had been cultivated aerobically than anaerobically. These are anaerobic and it's been cultivated under those conditions. And what we're seeing here are the urea plasma, the arrows pointing at them. And then we've got just cellular debris. So lots of that stuff that you need to kind of weed through to recognize the urea plasma. Also, feel like you guys did the urease tests and saw those tubes turning bright pink with Proteus, likely was the one that did that. I feel like these should do the same, but that's also, it would be a different situation because it'd have to be cultivated anaerobically and not just in those little slip cap tubes. So for some bacterial infections of the urinary tract, there are numerous players that can set the stage for issues. And like I said, in some instances, we might think of our own normal flora lending to some of these issues. Cystitis, handfuls of bacteria, E. coli at the top of the list, Enterococcus faecalis, Streptococcus algalactiae, which I believe this is the culprit for the puerperal or childbed fever. This guy right here, Klebsiella pneumonia, Staphylococcus saprophyticus, and some others can all result in cystitis. Dysuria, so problems urinating, pyuria, pus in the urine, hematuria would be blood, bladder pain, more common to see this occurring in females than in males, and part of it's shorter urethra, and lots of flora within the vagina that sets the stage for some of these potentially causing issues. This is non-transmissible. It's an opportunistic situation. Fecal bacteria introduced into the urinary tract. And if the immune response is depleted a little bit, it can set the stage for that. The colonies, culturing, and the urine dipstick helpful with that. So the leptospirosis, like I said, zoonotic, animals to humans, contact with urine or bodily fluids, fevers and headaches, chills and vomiting, all sorts of issues if it disseminates, lending to problems for sure. Non-gonococcal urethritis, so not gonorrhea, chlamydia, mycoplasma, a couple of different ones, and the urea plasma can all cause urethritis. Obviously, gonorrhea, that we will talk about in a second, can also cause this, but ruling out that, other players. Could be mild to asymptomatic, also potentially lending to purulent discharge and dysuria. This can be sexually transmitted. It can also be an infection transmitted in the process of delivery, setting the stage for the child to have issues. The pleonephritis and glomerulonephritis, like I said a second ago, pyogenes with those immune complexes can set the stage for this, but we can also see E. coli, Proteus, and Klebsiella also lending to that. Another non-transmissible urine flora getting inoculated into the urinary tract, or through the circulation, if we're thinking pyogenes. So shifting gears, this smear, I'm assuming pap smear is what we're looking at here. I don't think any sort of staining on here. I'm not seeing anything that looks colored, although it's greenish looking. All right. I don't think stain has been applied. We've got a couple of normal cells, cervical cells, and then we have a cell that looks very, very different. all right? And we call these clue cells. These are basically these cells that are riddled with bacteria all over them, all right? And we'll move through here in a second. Gardnerella is a culprit for this. We'll get to that. Now for the fun stuff. Such a sparse group. There's probably nobody even on Zoom. Nobody wanted this today, right? I could feel it. I knew you didn't want it Friday either. All right. So this image, we're looking at gonorrhea. Neisseria gonorrhoeae is a gram-negative coccus. That's what we're looking at in this image. This is full of neutrophils, all right? And typically when we're thinking neutrophils, I mean, we have gram stained these, the smear, so it's different than doing a blood smear. These neutrophils don't look like a neutrophil would look with a blood smear, all right? Here, I'm like, that looks more eosinophil-like, but the nuclei look different, all right? Eosinophils are more pinkish like this, all right? But with a gram stain, they would pick up the crystal or the safranin. There is no cell wall. They're going to show up as negatives, all right? But they really don't have a gram reaction. It's the little speckles in here. These are the Neisseria bacteria, and kind of zooming in on them, they look sort of rounded, kidney bean sort of shape, diplococci, two of them together, like Streptococcus pneumoniae, but pink, negative staining versus the strep that's a positive, all right? So we could see discharge in both sexes. It looks like potentially maybe something else going on. Maybe, all right. The lesions indicating another, right? Maybe syphilis. We'll get to that in a second, all right? So chlamydia, I know we have mentioned this previously. Chlamydia is a little bit different than most of our bacteria in the fact that it has to be inside of a cell to survive. So we call it an obligate intracellular bacterium or an obligate intracellular parasite. They call them that as well, but it's bacteria, all right? So here we can see a few normal cells in all surrounding, and a few, three of them, that have this darkish pigment within them. We call these McCoy cells, McCoy cell monolayers, and the chlamydia getting in there, all right? And they replicate, deplete all of the nutrients within that cell, destroying it, rupturing out, releasing more of these reticulate bodies to infect new cells, and it keeps going. Here we could see some swollen lymph nodes, lymphogranuloma venereum, as a result of chlamydial infection. So, and we call these buboes, bubonic plague, similar deal, but Yersinia pestis causing the swollen lymph nodes instead of chlamydia. So, next image is syphilis. A spirochete, Treponema pallidum, is the bacterium. There are three stages of syphilis, and we've got them diagrammed here. The primary stage, there's an ulcerated sore, a hard chancre, and this we will compare and contrast the littlest bit between syphilis and Haemophilus here in 1 second. Similar sorts of lesions, little bit, but with syphilis, these lesions are painless, although that looks like it should be very painful, all right? Painless with syphilis, painful with Haemophilus, all right? The primary. Secondary, we see secondary syphilis with rash symptoms, potentially starting on the palms. They can be throughout the body, but different, not ulcerated lesions that we see, and truly depends on whether or not antimicrobial therapy has been sought to prevent progressing to these latter stages. Tertiary syphilis, all sorts of potential lesions, gumas, these ulcers that you can see disseminated throughout the body, bone involvement, central nervous system involvement, all sorts of potentials with that, and potentially kind of lending to madness if we think about central nervous system situations. And there's all sorts of historical accounts of individuals that maybe were plagued with that and kind of lost their minds later in life. Much better to just see the bacteria, I feel like. Our spirochete right here, this is through some dark field microscopy, so they sort of look like they're glowing in the dark. And then another type of staining technique we can use with these spirochetes is silver staining. So you can see all of them, and then some of our own cells in the background, a little in yellow, but the spirochetes all over. There are other potentials with some immunological staining techniques where we can get antibodies linking up to the bacteria. And if they do link, we'll get some fluorescence. So immunofluorescence staining is another potential with that. So Haemophilus, as I said, these chancres are painful, all right? Haemophilus ducreyi is the bacterium. I'd like to say do cry because you're probably going to cry, right? These are gram-negative bacteria. You could see them all kind of look like sort of chained up, linked together. And this cultured in a rabbit blood agar plate. There's all sorts of different potentials for blood agar. Sheep and cow blood are more common, but I believe something, and I don't remember the details, but some sort of nutrient within the rabbit blood is what Haemophilus really likes for cultivation. And I also wonder if it's chocolate-like, if it's been cooked up, because most Haemophilus, I believe, can't lyse those cells, can't grow without those nutrients. So I'm not sure with that. All right, bacterial vaginosis, Gardnerella vaginalis, a main culprit with it. Bacteroides, Fusobacterium are also some potentials. This can be asymptomatic, but it can lend to recognition. Vaginal discharge, burning, odor, and itching are things that you would recognize as not being asymptomatic. This is an opportunistic, so these bacteria there as part of the normal flora, not the same as a yeast infection, but a similar sort of an idea where their populations end up growing more out of control, lending to these situations. So an imbalance of that normal vaginal microbiota, it can be hormone related that lends to some of these situations for vaginosis, vaginal smear, and some antimicrobials. So our chancroid Haemophilus, soft, painful chancres on the genitals, but they can be elsewhere, mouth, anus, all depends on sexual practice. We think about contact with the open lesions or the discharge that sets the stage for transmission. Also testing to determine, are we looking at somebody else, syphilis and herpes as well, because you can see some similarities with the lesions, and handful of antimicrobials. Chlamydia, this bacterium, as far as sexually transmitted infections go, I am pretty sure it is the top of the list. It is very common, which seems super disturbing. All right. And part of that lends to the fact that it may be asymptomatic. You may be infected with it and have no clue, and that sets the stage to share. So often asymptomatic, but if symptoms do result in men, we can see urethritis, epididymitis, orchitis. In women, urethritis again, discharge, bleeding, pelvic inflammatory disease. There's a handful that can stimulate this, sapropharyngitis, which I believe is swollen and inflamed fallopian tubes, if I remember properly. And it also sets the stage for increasing the risk of cervical cancer. So, sexual contact or in transit through the birth canal are opportunities for infection, respectively. Gonorrhea, Neisseria gonorrhoeae. We don't see anything up here that says asymptomatic. If you've got this, you're not asymptomatic. All right. Urethritis, dysuria, penile or vaginal discharge. We saw an image of that. Rectal pain and bleeding as a potential in females, pelvic pain, intramenstrual bleeding. So not at the right time. That's a potential flag. There are also situations with hormones that may lend to some of that. So not always like, pelvic inflammatory disease. This is definitely a serious one when we think about gonorrhea. Sapropharyngitis, infertility as a result of that, and the potential with the massive inflammation and scarring within the fallopian tubes can lend to ectopic pregnancies. So the fertilized egg isn't able to make a transit through the fallopian tube to the uterus, and it tries to develop ectopically. Bad news. There are potentials for dissemination, arthritis, endocarditis, and meningitis as well. So little of this will be discussed later. So sexually transmitted. Syphilis, as I said, multiple phases with it. Initially in the primary stage, the hard chancre, again, painless. Moving to the secondary phase, rash, cutaneous lesions, fever, lymph nodes, moving into tertiary, those ulcers, the gumas, cardiovascular, neurosyphilis, and potential fatal contact. So, enough of that. For virus, herpes has got to be right up there, top of the list when we think about some of these potentials. So here's a bunch of herpes virions. Herpes simplex is what we're saying here. And I think ultimately, looking through the electron microscope, you wouldn't know if we're talking type 1 or type 2. They are going to look very, very similar. It really just depends on where on the body we're talking about. So with genital herpes, typically thinking type 2, but type 1 can still manifest in a similar fashion and vice versa. So we could see these lesions, but they also may appear elsewhere and look very different, almost pimple-like. So skin or mucous membranes, all right? And this sets the stage for maybe overlooked and not recognized, lending to opportunities to spread to others. Human papilloma viruses, boatloads of different types of HPV virus, some of which set the stage for genital warts, anus or genitalia, or both, potentially impacted, and it truly depends on circumstances, sexual practice. Here, another pap smear, and what we are looking at initially normal cells, and the larger huge nuclei within here and enlarged cell itself are the infected cells. And these are the cells infected with HPV. The hyperchromasia, this swelling and enlarged nuclei are characteristic of that. And it looks as if we've potentially got some fusion of cells. Coleocytes are what we call these infected cells. So for our viral, cervical cancer, HPV, the human papillomavirus, 16 and 18 are the common types or strains of that, but there are others. There are potentials for cervical cancer, but it can also develop elsewhere, in the mouth, anus, all sorts of opportunities, not just cervical cancer. There are vaccines against this, Gardasil and Cervarix, to protect against the development of this. And we think about contact, direct contact to transmit. As I said, with genital herpes, herpes simplex 1 or 2, 2 being more common, recurring outbreaks on the skin vesicles of the genitalia or elsewhere. And there are potentials for asymptomatic situations in individuals where you don't recognize it, lending to opportunities to share. So human papillomaviruses, we've got some up above with the potential for cervical cancer, but there are others that are more just thinking wart, not cancer potentials. So still HPV, but some different strains of that. All right. So genital warts or warts elsewhere on your body. We already talked some warts when we were talking skin infection. All right. Same players. Neonatal herpes, this would be in transit through the birth canal or transplacentally in some instances where it's able to cross the placenta, infect before transit through the birth canal. This is more of a disseminated or systemic infection when we think about that as it's infecting the newborn or still cooking. Vesicles on the skin, mucous membranes, eyes. If it's disseminated situation, we think about serious complications, motor impairment, possible death of the fetus or the newborn. So we've talked about yeast infections repeatedly as we've gone through. It's a big deal when we think about antibiotics, especially some of those broad spectrum drugs. These yeast cells are part of our normal flora in small quantities. And when the situation is set for them to grow more out of control, all sorts of opportunities may present. And here, as we're thinking urinary tract, we're thinking yeast infection. All right. So images with our yeast cells staining up nicely and kind of this bluish. You can see the thick cell wall. There are some protrusions, the pseudohyphae that look like filamentous mold, but it's not truly that. It's just a projection, partially lending to some virulence with these yeast cells, allowing them to kind of anchor and tether themselves. And the blastospores, which is basically these cells as they are replicating, or what we're looking at with that. So here we can see the mother cell, the daughter cell. This is still our candida. They replicate by budding, and this germ tube, only 6 minutes old, they say, is this protrusion, the pseudohyphae that, sorry, wrong way, that we can see some of that right here, very protruding out. So, two different things we're looking at here. First off, we have a normal cell and a load of bacteria surrounding it. These bacteria are lactobacilli. There are numerous types of lactobacillus that are normal flora in the vagina. You want these there. And when their numbers become depleted, the image next door is the potential. So this is the Candida albicans, arrow pointing to some of the pseudohyphae, but you can see a few of the yeast cells. It's really kind of difficult to see very well. This looks similar to that other image that was kind of greenish. And this one, not stained, but reagent has been applied, potassium hydroxide, call it a KOH prep, and it helps to kind of break down like some of the keratin within the cells, lending to the potentials to maybe visualize things a little better. So this helps to clear away our own cells, and what's left behind are the yeast is really what we're seeing, all right? So the lactobacillus creates a habitat within the vagina that becomes more acidic, which makes it pretty inhospitable to numerous types of organisms, all right? Helping to create a habitat that's not conducive to their colonization, all right? And as they get depleted, yeast cells and other organisms potentially may have an opportunity to take hold. And another reason with antibiotic therapy that the consumption of yogurt helps to mitigate the depletion of these within the vagina. Next is a type of protozoal parasite, Trichomonas vaginalis. This organism, when it comes to protists, lots of them have a vegetative stage, a trophozoite stage, and they also have a cyst stage. And the cysts are the typical players that infect, whether it's inoculation or consumption of that cyst, whatever it is. That's how you get infected. With Trichomonas vaginalis, it's only the trophozoite stage, the active swimming stage. There are no cyst stages. So it's direct transmission of this parasite. Sexual contact is typically what we think about for the transmission of Trichomonas vaginalis, but it can be transmitted other ways. Dirty washcloths, sharing towels, trying on swimsuits potentially sets the stage. That's why there's the little sticker in there that hopefully helps to protect, apparently. All right. I find that a bit disturbing. All right. If you're trying on swimsuits, you probably ought to leave your underwear on at the same time. That's what I feel like, right? An extra layer of protection, right? Don't know. You're like, I'm just order it off of Amazon, right? I don't know. So, fungal and protozoal, so eukaryotic. Trichomoniasis, or Trichomonas vaginalis, and then vaginal candidiasis or yeast infection. So, potentials with this, with our trichomoniasis, typically females are the ones that exhibit symptoms, and males that become infected oftentimes are asymptomatic, but not always. All right. Truly, recognition of this, both partners need to be treated. All right. And if not, the infection will come back. All right. Urethritis, vaginal or penile discharge, redness and soreness of the female genitalia. Like I said, oftentimes males don't recognize it. All right. A couple of azole drugs for treatment with it, and other azoles and some others for yeast infections, fluconazole, nystatin, and others. Topical application or pills. All right. Yay. how did we back? Nope. All right, quickly, let me introduce some of the GI tract. This is like I said, respiratory and GI are some of the main, really thinking clinical stuff, some of the main infections that I think you will be faced with when you're thinking about this. Uh, Not that STIs aren't something that we could observe as well. When I think about GI issues, oftentimes E. coli is one of the first things that pops into my mind, and I feel like it's nice that that's right who we start with in this chapter. I remember many years ago, I was pretty young, hearing about some E. coli outbreaks, and it was some of the, my first hints of hearing about this with a hamburger place that was dishing out some burgers that weren't quite cooked all of the way. There was some meat that had been contaminated, and handfuls of people, many handfuls of people got ill, and a few people died. All right. And this was one of the first instances of a big recall, and meat, burger especially, was what you heard about for a handful of these things. And then it slowly seemed to progress to all sorts of other things, vegetables that potentially could get you as well. It's not just hamburger. So the vegetarians, watch out, right? You can still get it, all right? Disturbing, all right? There was issue handful of years back, probably a decade, really. That's not a handful. Was some spinach that had E. coli. There was a child right down the road in American Falls that got sick by consuming some of the spinach. I think he was maybe 2 years old. He was pretty little, and he did not make it. All right. E. coli in little dudes or in elderly, huge issues. Not that I would want to get 0157 and just see what's up, right? Uh, Bad news all around. But as far as fatal infections go, they're definitely more susceptible. So there's a boatload of different strains of E. coli. The E. coli growing in our gut is called K12. It's something that we think of as really non pathogenic, but I feel like if you were to consume a little of that, you're probably still going to get the runs. All right. But it's not going to kill you. All right. 0157 maybe will. There are numerous things, toxins that can be secreted, different factors that may be released from these organisms, stimulating all sorts of pathogenicity in the host. Bloody diarrhea, a common one with 0157. All right. So we've got two images of our E. coli, and next door, all of this sticky looking stuff, these curly fibers. This is a virulence factor that 0157 harbors, like Velcro. All right. It helps them to adhere to surfaces, starts the formation of biofilms, and lends to a greater level of pathogenicity as a result of that. So, digestive tract, we are basically just a big tube with appendages to shovel food into that tube, from our mouth to our anus, all sorts of opportunities for organisms to potentially call it home, or maybe not. All right. Just depends. We've talked a little bit of the upper tract with respiratory, but it also fits with the GI tract. All right. So numerous things with potentials. All right. And I'm not going to go through all of those details. So, oh, my dental hygiene people, so little to talk about, but so much going on. All right. Uh, a small smattering that we get here. So, this a healthy situation. We've got our tooth, the roots with the enamel on here, providing protection to the inside, the business of your tooth. You can see healthy gums right here, going right up around the base of your tooth, covering that enamel. All right. Inside under the enamel, the dentin, the pulp cavity. And as we move into that, we've got nerves and blood vessels within. And when we start thinking about dental caries or cavities, we start moving into these areas. And that's when we potentially recognize issues. All right. And there are numerous things we will think about as far as bacteria causing issues as we progress through, but not a lot of detail, unfortunately. We'll get back to a little of that shortly. So moving down into the intestines, looking at the small intestine, lots of villi or macrovilli on these cells, increasing the absorptive area, enhancing our body's abilities to get as many nutrients in as possible. That's what we're looking at in this cross section right here. A blow up of the macrovilli within there. All right. As we look down below, we can see a cross section of that. The lumen of the intestine, where the foodstuffs are being digested, would be right here, and all of these cells absorbing. There's also goblet cells that are secreting. You can see that right here, providing the mucus secretions. They're within this as well. All right. So multiple, just enhancing, zooming in on it as we move through to see the individual microvilli with that. So same idea, cross section of this. So the intestinal cells, this is the lumen right here where the foodstuffs would be, microvilli and goblet cells surrounding this. All right. And how are all of these cells linked together right here? Tight junctions. Yes. And it comes full circle. All right. So with tooth decay, we've got all sorts of stuff going on here. All right. Bacterial biofilms, plaque, start to develop. Streptococcus mutans, we will see in a second, is a big player in this. We used to do some labs where we would look at body stuff, and one of them was this, trying to see, are you harboring mutans in your mouth or not? I can't remember the name of the plate. I want to say it was Snyder's auger, maybe. You would spit onto the plate, and you needed kind of a good amount of spit, a copious amount of spit onto the plate. This was probably the only plate that gets incubated upright. Everybody else we flip upside down. These plates, you spit on there and you just put the lid on and leave it. All right. And if you have this bacterium as it grows for a handful of days, the colonies are blackish and all raised up. They describe it in lab manuals like a kind of burnt sugar looking appearance. Not that you want to burn sugar. It reminded me of 4th of July stuff with the little snakes that you light. And right as you light that snake, just as it starts to grow the littlest bit, that is how it looks. Blackish, raised up, all weird. All right. So burnt sugar or snakes, right? Firework snakes, which I think is just a glob of carbon that's burning, really. I don't know. All right. So kind of interesting. And COVID, we stopped doing all of that. And after we stopped it, we kind of got together and we're like, is that a lot? You know, do we actually want the body stuff in the first place? I mean, do we want all the spit? Do we want the other areas as well, right? Because we opened up all sorts of opportunities, maybe some urine, maybe some feces. Want to see what's going on down there? Maybe you do, maybe you don't, right? Some did. Some took one for the team, right? Some of you, I could feel you're like, yes, right? I walked into lab and somebody had taken it on themselves with their swab to write poo on the plate. And E. coli, you know, turns pink as it's growing on a McConkey's auger plate. So pink poo written right on there. I was like, awesome. That is super cool, right? There you go. All right. Now you're all like, geez, couldn't we do that? I think your lab's done, right? You don't even have lab this week. Is that true? Dang it. All right. So enough of that. Our decay, I'll quickly hit this and then we will quit. So there are multiple opportunities as these bacteria colonize and start the biofilm formation. They start fermenting. We create more of a kind of anaerobic environment under this biofilm formation. Fermentation starts kicking into gear and we produce lots of acid. That acid starts to break through the enamel lining of the tooth. That's our decay that we see up above as it's starting to bore through there. As we make our way into the dentin and then in through the pulp as we pour all of the vessels and all of that is the potential for some serious pain. Progressing deeper, we can see some abscesses potentially forming, also maybe lending to the loss of a tooth. There's multiple things we will talk about with eroding gums and all of that on Wednesday. All right, I will quit with that. Farewell, good day, brush your teeth and wash your hands. Wash your hands, then brush your teeth.